All right, thank you everybody for coming to the Center for Evolution and Medicine seminar today. I am beyond thrilled to get to introduce Lee Nelson to you. So the first fun fact is that we found out she and Dr. Andy Nessie share some relatives in their pedigree in Norway. I do not have such a strong connection to her, but I have been a huge science fangirl of her work, and she has been really instrumental in studying a process that I find fascinating, which is microchimerism, that she'll talk to you more about today. But as her brief introduction, I'm trying to stand still, but it's really fantastic to get to invite her here. I know scientists are people. But there are people whose work is just really amazing. And so when you get to meet one of your science idols, it's nerve-wracking. So Lee got her bachelor's at Stanford University in philosophy and did her MD at the University of California, Davis, which I'm excited to go visit in a few months. I've never been there, but I've heard it's a lovely place. And she did some postdoctoral trainings before settling at the University of Washington in 1981. And since then has been in the rainy state. And last night she said that the rain didn't bother her for the first 15 years, but it's starting to become a little old. So I'm very excited that we get to have her out here to the sunny Arizona, and hopefully we'll get to have her out a little bit more. She has a couple patents and has been PI on about 30 grants, and has approximately 129 publications. So she will give us some snapshots of the work that she's been doing today. If you are interested, there's a little bit of time this afternoon if you didn't get on the schedule. So I'm happy to talk with you after the seminar about that. And without further ado, let's introduce our speaker. Welcome our speaker.
It's great to be here. Thank you for the invitation. Thank you for the sunshine. So what I am going to talk about is bidirectional fetal-maternal cell exchange and DNA exchange that occurs during normal pregnancy. And microchimerism then refers to a small number of cells or DNA that derive from a genetically disparate individuals. So this term comes from the mythological chimera here, who is part lion and part goat and part serpent. But in medicine then we're referring to cells or DNA that came from a different person, and micro just meaning small. So I think the big surprise was that it turns out there must be some cellular exchange, because 10, 20, 30 years later, you can find cells that were of fetal origin in women who have given birth, and cells of maternal origin in the rest of us. So what I'm going to attempt to do here is give you just a little bit of history and then try to give you the big picture view. Because we started out working in autoimmunity here. My background is actually as a rheumatologist, and we get a lot of the autoimmune diseases. But I used to go around talking in the early years about this phenomenon and say if I were smarter, I would study some of the other broad implications of this phenomena. And so I actually started doing that. And other people have done that as well. So I'm going to try to give you a snapshot of some of the other areas where this naturally acquired microchimerism, of which the primary source then is this maternal-fetal exchange, has impact in development, cancer, regeneration, and thanks to the folks here in evolution, and certainly in reproduction. So I started out on this disease because it's a really mean disease and we really don't treat it very well. And it's called systemic sclerosis. It has this other name of scleroderma, which means thickened and then skin. And that's exactly what happens. So you can see somebody is not able to actually even extend out their fingers, not because there's something wrong with their joints, but because the skin is just so thickened. And it actually results in breakdown that can be happen, they can actually lose digits because of the breakdown in the skin. But really the big problem is it moves to the internal organs. So here's an x-ray showing the lung, which should be nice and dark colored with lots of space where you can move air, but instead is whitened out. So this actually is a disease that has a prognosis in its more serious form that's worse than cancer, and we really don't treat it very well. So one day a friend, actually a colleague of mine, called me up and he said that somebody out at a company had tested a female technician who had delivered a son a year earlier and that there was still male DNA in her blood. And it was like a light bulb went off. And so I came up with this really radical notion at the time that maybe some of these autoimmune diseases aren't entirely autoimmune. So this word allo just means another individual in your same species, but another individual. So I submitted this as actually a peer-reviewed hypothesis paper with no data, which people generally don't do, and asked this question, whether some autoimmune disease is actually auto-alloimmune or allo-autoimmune, and then set about to study this particular disease, which is predominant in women, and it has a peak incidence in post-reproductive years, and my lab is based at a transplant center where bone marrow, used to be called bone marrow transplant, was kind of pioneered by Don Thomas, who shared a Nobel Prize for that. And that bone marrow transplant results in a picture that looks a lot like this disease. So that was kind of the other observation that made me propose this radical hypothesis. So what we did is we collaborated with Diana Bianchi's group on the East Coast and a company that at the time all that could be done was to use a prenatal diagnosis test to do a semi-quantitative analysis of male DNA in the blood. And we did this prospective blinded study. We picked women who had given birth to sons and we tested the DNA then with their assay extracted from whole peripheral blood. And so this was the first description of microchimerism in an autoimmune disease. When we broke the code, there was more prevalent, but more strikingly higher quantities of male DNA. This is in a tablespoon of blood, more than 60 male cell equivalents in a tablespoon, and the woman had given birth to her son 20, 30 years earlier. So that was part of the story. But the other part of the story in that paper, in my mind, was actually more important because it speaks more directly to something that can't really be an epiphenomena, that these cells just traveled there after the disease started. And that was, we looked at the HLA relationship of the woman and her previously born child. And by the way, I should backtrack just a moment and say the reason we studied women with sons was not because we thought there was something special about having a son. It's because that was the only assay that was available at the time. So presumably the same thing would be the case with daughters. So these are the set of genes on the human 6th chromosome here in the short arm that are typed for if somebody needs a transplant. So they're the primary genes that determine if you'll accept or reject a transplant and whether you get this graft versus host complication. The classical ones that are typed for are A, B, and C, and then DR and DQ and DP. And then that's defining this is me and this is something that would look foreign or look dangerous. And if you're an autoimmune oriented person, you're particularly interested in DR and DQ because it's known that particular variant forms of these genes give you susceptibility to just about every autoimmune disease that's been described. They're different ones for each disease, but each of the autoimmune diseases has an association with these class 2 genes. So for those of you who don't work in HLA, sometimes, particularly after lunch, people go, oh no, and they're just gone, because it seems complicated, but really it's not actually that complicated. What makes it complicated is the terminology. What happened is DNA typing wasn't available, and so the terminology began to develop here when people got together and exchanged antibodies, and that's how they defined HLA. And so they called something DR4. And then when DNA typing came in, they were able to actually discover that this gene encoding for this thing called DR4, actually there wasn't one of them, there were more than 50 of them. So there are a whole bunch of different variants. So this is really just a kind of splitting reductionist-like principle here. And now there are clearly more than 50 different variant forms of this. And so they named it with this. This is the gene you're looking at. This lets you know it's DNA typing. This lets you know that it's a 4, and then at O1, O2, O3, O54. And usually the ones that have the later numbers are ones that are less common. Not always, but usually. So everybody's used to this concept of genetic susceptibility, but actually what I wanted to look at was not denying that. That's a piece of the puzzle, but I wanted to add another piece of the puzzle, which was the HLA relationship of the mother to the child, just like you would for a donor and a recipient pair if somebody's coming for transplant. Now, I'm interested in the mother who got scleroderma, and so my hypothesis was that if she couldn't recognize the cell of the child as being foreign and different, that's when there might be problems. That might seem counterintuitive, but actually it's more like as if the cell could disrupt the mother's immune system regulation. And even though in, for example, this case, they're identical, that's only one of these HLA loci, okay? So most of the time what's gonna happen, this is the usual situation, the child inherits a form of HLA from the dad that differs from both of the mothers. So they're bidirectionally mismatched. Occasionally they'll be identical, such as here, and occasionally the child will inherit the same form of the gene from the father as from the mother. So that's called homozygosity. And I was particularly interested in this because in this situation, you can see the mother wouldn't see the child cell as foreign, but the child would see the mother's body as foreign. And of course, the reverse can happen. So it's really these two that we were interested in looking at. So the other half of that paper studied women who had developed scleroderma in healthy women. And indeed, we found that there's about a 9-fold increased chance that a woman would develop scleroderma if she had given birth to a child previously that had one of those two relationships with HLA to herself. And this is the kind of low-level resolution level like that DR4 you saw, and this is like the high-resolution level. So either way, there was an increased risk. And I say that really carries more weight because that relationship obviously preceded the woman getting scleroderma, whereas you could always argue that, well, maybe these cells actually traveled there secondarily after the disease. But then we were face-to-face with the second step. Now, any of you working in the lab for any period of time have probably had this experience. I just saw it on a greeting card and I said, yeah, this is really what it feels like. Because you can look for male DNA or male cells in a female, but what are we going to do to look at maternal cells in a woman or daughter cells in a woman? And so we had to develop an approach to do this. Well, one thing you can do, you could pick a boy to look, or a male, to look for female cells and put fluorescent probes on the X and Y chromosome and carefully look for the ones that are XX. But this would be a very tedious way forward, and it would be lots of time in a darkroom, so it's really not practical. So what we did is that we developed this method by which we would target what was unique in the DNA on an HLA gene of, for example, a mother in this particular illustration. So here we could take either a daughter or a son, and we could say, okay, this daughter or son did not inherit B44. So now we're going to look and we're going to test the daughter or son and see if we see B44 anyway. So that involves designing primers and then specific probes and then proving that your system is accurate. And so then over time, we developed an extensive panel of quantitative PCR assays that target all these HLA specificities. So when we developed the first set of those assays, Natalie Lambert in the lab tested women with scleroderma and healthy women to see, is there also an increase in maternal microchimerism? And this was less striking than the microchimerism of fetal origin, but it was significant that there was more maternal microchimerism in these scleroderma patients than in the controls. Ah, yes, my cat. So the reason my cat is here is not because this is a long tail, like his long tail that he was proud of. It's actually a very short tail. So what was happening, he describes the next part of the story the best. So we came home after a remodel to discover that the contractors had cut about a 12-foot by 12-foot hole on the north side of our house. And then on the south side of the house, they had cut another hole that was equally as big. And so my husband turned to me and he said, "Does it strike you that we have an animal security problem here?" And I said, "Well, yeah, it does, but I mean, what are we going to do? You know, we're not going to check into a hotel." So we went to sleep that night in our bed, and this is a true story. And at 3 a.m., we were awakened by a cat fight under our bed. And so we each got up, and we each looked under the bed, and neither cat was our cat. And so, admittedly, I was a little sleep deprived the next morning when I went into work, but I said to Natalie Lambert, who's French, I said, "Well, you know, if you've got maternal cells on the one side and then you've got these fetal origin cells on the other side, I mean, you know, could there actually be something kind of across generations?" Well, I told you Natalie was French, and so without missing a beat, she said, "Well, you know, of course." And this is actually what Rodin's thinker must have been pondering all this time. And so we then asked those questions. Can an individual harbor multi-generational microchimerism, a woman? And how about HLA compatibility across the generations? I think this ties in nicely to the evolutionary themes. So I'm answering the second part of that question first, and this is not published yet for a variety of reasons. But this is looking across generations. So essentially, you've got the person I'm concerned about is the woman in the middle, but this is looking at her child and her mother. So essentially, the grandchild's relationship to his grandmother, okay? And what it looks like is that if the grandmother is not distinguishable from the point of view of the grandchild, there's a significant increased risk of this disease. And again, this is across class 2. And I forgot to tell you with the other first part of this story that class 1 doesn't make any difference. So that's also very interesting, since class 2 is where the genes that are associated with the disease happen. And these are genes that travel along with the DR gene, and with the most striking finding at DQA1. And by the way, because what this really has to say is the child's relationship to the grandmother, and it's what's not transmitted. This is essentially a story about a man and his mother-in-law. So you got to watch that too in your choice of mates. The second direct answer to is there microchimerism across generations is best illustrated here in an autopsy study. This is a woman that was part of my study. So I had her whole family typed, and then she got very, very ill and came to get what's called an autologous transplant, where they take your own cells out and then give them back to you and hope to reset the autoimmune regulatory mechanisms. But sadly, she died. And that, though, enabled us to actually look for both maternal microchimerism quantitatively with this panel and the fetal origin microchimerism. So you can see this is expressed as the number of microchimeric cell equivalents per million tested, that they're really very high levels in the lung, which is how we lose our patients most of the time, but also pretty high levels of both types in the heart. Some sequestering in the spleen, which has also been suggested for at least the fetal microchimerism in another study. And then a little bit of a reverse direction with a little more maternal in pancreas and in bone marrow. But the more striking thing is actually the quantities. Clearly, we do have multi-generational microchimerism. It may not actually be reflected in peripheral blood. Most of the time, her peripheral blood was actually negative. But clearly, in these organs, these are high levels. So now, in order to get back to the next step of the scleroderma story, I have to tell you about other diseases, because it's in other diseases that we're able to fill in the next piece of the story. So these are just a few of the autoimmune diseases that have now been investigated for microchimerism. And the one I want to talk about is neonatal lupus, because it kind of gives you the next big picture view of kind of the conceptual advance. This was done by a pediatric rheumatologist, Ann Stevens, in my lab, who was interested in this disease called neonatal lupus. So you can see this baby has a rash. It's a really interesting disease because the mothers, sometimes they have lupus, but most of the time the mothers are actually well. but they have these certain autoantibodies, and when those are transferred to the fetus can develop this neonatal lupus syndrome. And the most serious part of it is this congenital heart block, which used to result before we could identify this and do interventions in a very high mortality rate. So Anne asked the question, is maternal microchimerism involved in the pathogenesis of this heart block that occurs in these babies? And what she did is went and got autopsy samples on boy babies so she could look for XX cells in an XY boy. I still remember when we first looked at this in our lab meeting. This is the nucleus right here in blue, and here's an XX female cell, and mostly XY cells. We looked at this and just said, boy, that looks like that's really almost integrated right in with the heart tissue. And so then she spent quite a while trying to develop a technique where she could actually do this fluorescent technique, so she knew the cell was from the mother, presumably a female cell, but also know what kind of cell is it. And after a lot of work on that, she got it to work and found that this is the exact same cell here. Here's a female cell, that 80% of these cells expressed sarcomeric alpha-actin. So that means they're actually cardiac myocytes. So the cells that are exchanged and that we acquire and that we have long term aren't just circulating blood cells, they're actually part of the body architecture. I kind of visualize it a little bit like, just like a little snowflake of cells that you get that are both circulatory but also part of different organs, and that's Anne. She looks set actually for the Arizona sunshine there. So at this point in time, we actually know that there are a variety of different cell types besides blood cells in humans. So this is a summary of what's been found in humans, at least what was found as of 2012. There's a few more added since then. So you can see you can find cardiac myocytes of both types and hepatocytes of both types, and a variety of others, beta islet cells that are actually female, presumably maternal, et cetera. And then I couldn't resist, though, keeping on one experimental study here, which is this one that showed that the fetal cells can actually become neurons in the mother's brain. I don't know how people feel about that, but there you are. And it's a very nice study out of Singapore. So how might these cells be involved in autoimmune disease, for better or for worse? I think in the beginning, people thought, because they were unfamiliar with this, they must be bad. And so they thought they must be attacking my normal tissue. And that actually isn't my first thought, even though this disease looks like graft-versus-host disease, partly because graft-versus-host disease, 100% of the cells are from the donor. And here we're talking about less than 1%. But a way in which these cells could really cause problems is what's called indirect antigen presentation. So this is a case where it's actually a host cell to host cell. So it's your cell presenting to your own cell, but it's presenting a peptide from this microchimeric cell. And that could actually disrupt the immune regulatory networks. And people in particularly solid organ transplant are very familiar with this pathway. And then there's this more radical hypothesis that really comes up after you saw the neonatal lupus work, which is, well, okay, maybe we should just change our concept of who we are, recognize that we're actually more diverse than we thought we were, and that these cells are part of us. And if these differentiated cells, including the maternal microchimerism, is part of normal biology, then you could actually get an autoimmune disease by loss of tolerance. So it's kind of turning autoimmunity a little bit on its head. And that's what we think actually happens in neonatal lupus. Not in all the autoimmune diseases, but in that disease, at least that's what we would hypothesize. But there's the other side of that, which is these are common in normal healthy people. So what's going on in normal healthy people? Well, it's now been shown in two very nice studies that the fetus produces regulatory cells to the mother that endure. So obviously we aren't invested in just getting rid of these cells that we acquire from our mother. And the same thing occurs in the other direction. The mother produces T regulatory cells to the fetus, and those also endure. And now this is studies in humans here. And then I couldn't help but include mention of this really nice article by Samstein et al., where they actually showed that placental mammals have a FOXP3 enhancer that's essential for peripheral T regulatory cells, but not for thymic T regulatory cells, because I thought this pertained to the evolution theme. And you can imagine that the cells that you acquire from your mother, you're acquiring them when you're defining who you are. And so the immunology of that is likely to be very different and kind of central. Whereas the cells that a woman acquires after a pregnancy are actually being acquired to an immune system that's already developed and thinks it knows what self is. So the immunology is likely to be quite different and very interesting when we figure that out. So this brings me to the more simple expression of this, which was this colleague, friend, who was a retired professor who, out of the blue, sent me an email, and we developed this kind of intellectual love affair. And I've since met with him a couple times. He's come to visit me, and I've gone to visit him. But one day this email pops up on my screen, and it says, and I can't do the British accent, I wish I could to do this, but I could kind of hear it. Perhaps you should consider the placenta as a selective immigration policy. And I thought, boy, he's got this nailed, you know. What an easy way to express it. I think that's exactly the right model. Because then you can see the maternal cells are like, that's what, when the nation was actually forming, they were already there. But then you can imagine an immigrant who arrives 20, 30 years later is going to have a little bit of accent, is still going to want, you know, the tea made a certain way that's a little bit different, and yada, yada. So I thought that was well put. So then Hilary Gamble came along, and she's maternal-fetal medicine physician scientist. And she said, okay, what happens when a woman gets pregnant? She's already got a graft of these immigrant cells from her mother, and now she gets a new set of immigrants from her son or her daughter. Do they compete in the same way that we know when in the setting of transplant, when you do two cord bloods, they compete. And even though the outcome is better, one of them wins 90% of the time. So she's asking that similar kind of a question. So she studied women who had had 1 to 4 births, and this is studying peripheral blood again. So at least on the peripheral blood level, the results suggested that indeed that is what's happening, that when you've had multiple pregnancies, births at least, that those cells will actually kick out the other graft. So this would be like the grandchild saying, okay, grandma, you did your job, but you know, now I'm here and I'm younger and I'm better. And maybe this is one of the things that contributes to female longevity. I don't know, but it's worth considering. So with those pieces put together, I can tell you where we are with what we think is going on in the disease that I would most like to see there be a therapeutic change for. So, and that is scleroderma. So what I think, this is all hypothesis, is that maternal microchimerism is acquired normally during fetal life, and it produces differentiated maternal microchimerism in tissues, like you've seen, maybe, you know, a few beta islet cells, a few little heart cells, this, that, and the other thing. And then normally, when you get the new graft with fetal microchimerism, those are displaced in tissues and in blood. But when you have this kind of HLA situation across the generations, where the fetal microchimerism or the fetal origin microchimerism can't see the maternal, this is the maternal to the woman, okay? We often use the word proband so we don't get confused about which mother we're talking about, but I think you understand we're talking across generations here, is indistinguishable from the fetal perspective, then that normal replacement can't happen. And yet, the cells of the woman's mother can see those cells of the child as being foreign. And so you have this indolent inflammation, which is actually what this disease looks like, is indolent inflammation. So the first part of testing that hypothesis is, are there vascular endothelial cells in the lung of scleroderma patients that are actually chimeric? And so this also isn't published yet. This is the work of Colleen Gentile. And it's actually surprised me. I figured we'd be wrong, because usually I'm wrong, but I figure out something interesting along the way. But in this particular case so far, it looks like we looked at a male first, because that way we could look for XX female cells, and here are 3, and also stain them by immunohistochemistry. And indeed, there are actually vascular endothelial cells that are maternal. And they're actually quite high in this one patient. And now we're working on 7 other autopsies to try to figure out that piece of the puzzle. So that's a story on scleroderma. And so I'm going to switch gears to rheumatoid arthritis because it illustrates another important point, but a different point. It's also a great disease for showing both the good and the bad of microchimerism. So most of you will probably be familiar with this. So even though scleroderma is a rare disease, this is not a rare disease. It's got a 1% incidence. It's also got a female to male increased ratio. And it's got this hallmark figure of symmetrical polyarthritis that is potentially very destructive and disabling and deforming. So it's known that there's an HLA class II sequence that's associated with this disease. And that sequence is from position 70 to 74 of the HLA DR beta 1 chain. It's the one that encodes that DR molecule. And it's all of these sequences in the red box. These guys here are all positively charged, and each of them is associated. So it's got a special name, the sequence called the shared epitope. And it's very clear that that's the susceptibility and far and away and above stronger than anything that's come up in the genome-wide scans that wasn't HLA sensitive. Additionally, there's a sequence that's protective. And this is negatively charged. Both of these are negatively charged. And they're strongly protective against getting rheumatoid arthritis. By the way, I have 2 of them. This is good. So here we have that 0402, which is the protective one. And what we want to do is we want to look at whether the non-inherited gene from the mother has that protective sequence. So by inference, we're saying, okay, we also think there's maternal microchimerism that has this sequence. But even if you just look at the HLA, you look at what was not inherited, because that's what you would have by microchimerism. And so this has a term, called NEMA, which is popular in the transplant community. This is from a group in the Netherlands. They studied 2 different populations, and in both of them, they confirmed that indeed, in rheumatoid arthritis patients, there's a deficiency in having that sequence from the mother versus the father, non-inherited. So very strongly pointing the finger at your mom failed to protect you by giving you one of these protective sequences. So that's the good. Well, Natalie by now had relocated herself to her own lab in Marseille, and I highly recommend going there to postdoc with her. I would go to work for her myself. Fabulous mentor. And she said, well, I'm going to look at the other side of this. I'm going to look at the bad side. What if there's this 20 to 30% of people with rheumatoid arthritis that don't have any genetic risk? Maybe they got it by microchimerism. Maybe there's this mini gene transfer of pregnancy. Why not? And so she took women in the French population that didn't have the rheumatoid arthritis risk gene, which is usually on a DR4, and healthy controls, and studied them with one of these qPCR assays that we had developed. And indeed, the rheumatoid arthritis patients had it by microchimerism. So they had a mini gene transfer of the susceptibility, presumably the susceptibility gene. Her study actually looked at DR4, and not all DR4 is the susceptibility. One of them is actually the protective one. We actually did a subsequent study that looked at the exact sequence and extended and confirmed that. So it still holds up, and you don't see it really much in controls. And some of these are actually pretty high levels. Some of them are above the microchimerism level up here at 5%. So more simply stated, transgenerational microchimerism in women with a potential for both good or bad. So this is why I'm using my own computer so that this animation will work for you, because we wanted to kind of put this in real time. So we're looking at the transfer cells initially in the first generation. And then if the fetus here is a girl, and she grows up, and you can see the maternal cells in her. And then she has her own pregnancy. And then if you watch this, you'll see some of those maternal cells actually disappear. And then if you just add to that, to remember that it's not just these circulating blood cells, but we're also talking about a small population of cells in the heart, in the pancreas, in the tubule of the kidney. Then you kind of have a real-time picture. So now I have to complicate it just a little bit more to say that there are other ways you can get microchimerism. So you may be asking, well, what about men and what about children? One way is twin-twin transfer. That was described in cows a long time ago, but it actually has been shown since that it happens in humans. And the obstetricians tell us that actually a fair number of pregnancies begin as twins, and there's a vanished twin. So you actually might become chimeric from a twin you didn't know that you had. Another possibility is from an older sibling. It hasn't really been definitively shown. I don't think it probably happens very often, but it certainly could happen because your mother is hanging onto the cells from you in a small amount, and then she gets pregnant with your younger brother or sister. Occasionally, you might get a stem cell and it might engraft from an older brother or sister. And kind of underscoring the fact that I think that these sources must be happening and we just haven't quite figured out how often it is this, what I think was a very nice study done by a French group showing that even if you look in a fetus liver of a female, you can find XY cells. And in this case, this is actually a hepatocyte, so a male hepatocyte in a female fetus liver. So I think the story is complicated, more complex, and there are more ways that you can get multi-sources of microchimerism. So in the remaining time, I just want to give a little snapshot of what are very important other areas for naturally acquired microchimerism: cancer, regeneration, and evolution. So in cancer, what we did is we started with this observation that if you get a transplant, because as I said, I'm located at a transplant center for something like leukemia or lymphoma, if you have a mismatch and get graft versus host disease, well, that's not fun because it's got problems, but you're less likely to recur with your cancer. And so by that principle, wouldn't the same hold for naturally acquired microchimerism, which is also going to have HLA differences? And in particular, it's known that having had a birth reduces your risk for a while of getting breast cancer. So V.K. Gotti is an oncologist, MD, PhD, who came to work with me, and he asked that question. He says, does HLA disparate microchimerism of fetal origin contribute to this known observation about how having had a birth protects against breast cancer? So he since did a number of studies, and you don't need all the detail, but just to know that it was consistently shown across 3 different studies that he did that there's a deficiency in fetal origin microchimerism in women with breast cancer. This isn't the only study. There's another one that confirmed it, and then there's another one that actually looked at breast cancer during pregnancy that may be a little bit different. But that's kind of the beginning of the cancer story and the part that we contributed here. But I have to say, this is now really its whole own field. And if anybody wants to read up on it, there's a very nice review by Fugazola. I think it's in Nature Endocrinology and in 2011 that talks about the different cancers. And again, the principle is both good and bad, because with a melanoma, it's thought that the fetal cells actually become vascular endothelial cells, so they actually fuel the growth of a melanoma. But most of the studies suggest that they're protective. And most of the parity effects are also protective. The same thing in the repair literature, not nearly as robust yet, and most of it is experimental. It's hard to demonstrate this kind of thing in humans, but this is a very lovely study by Hina Chadre's group that actually showed beating cardiac myocytes that were of fetal origin that could appear to be helping damage a maternal cardiac problem that arose during pregnancy. So I think there's substantial evidence also suggesting that these can be reparative. And then, most importantly here, is I'm just thrilled to see this article come out where somebody really finally, looked from an evolutionary perspective. And I sat on my couch and I read it all, and I looked up at my husband and I said, this is good. This is really good. And one of the things that really intrigued me was this hypothesis that maybe the fetal cells would particularly want to sequester in brain, thyroid, and breast, kind of areas where the maternal resources could be, kind of inspired to give more to the well-being of the child. And so I don't have the answers to that, but these are testable hypotheses. I have the answer to one of them, which is the brain. And this is work done by William Chan, who was a PhD in the lab. So he looked in autopsied human female brain, and he found that, and this happens to be women with no neurologic disease and also some women with Alzheimer's disease, that having male DNA was actually very common. So it was more than 50% of the time you could actually find male DNA in female brain. So presumably that's usually from prior pregnancy. I have to make reservations with this just because, I mean, it is autopsied material, and even though you're really careful about how you handle things against contamination, and I think the bank resources that handled these are also there. You're not quite sure, so I don't think you can use this as an exact prevalence, but it certainly tells you it's not uncommon that you have male DNA in female brain. So we're working, by the way, on the other side of that to look at maternal microchimerism in the brain, but we don't know all the answers yet. And this was William's first fish picture, and then he finished his postdoc and left me. So I still don't know what kind of cells these are, and it's driving me crazy. So we're going to try to figure that out. And then I just wanted to point out that there would be potential problems if evolution fast-tracked. And I saw that little picture, so sorry about that. That's what I thought of when I saw it. One of my own major interests in this area is the major histocompatibility complex, in other words, in humans, HLA, because these are obviously the genes that are so important in defining what's me versus what's something that's foreign or what's something that's dangerous. They're also involved so importantly in infectious disease and in cancer surveillance. And so this is just a huge topic. And it's one that I think is really going to be fascinating for hopefully some of you to help move us forward in our understanding. But, oh, and by the way, this guy here, you may wonder why I have this here. So this is a Picasso, actually, sculpture. And I think I saw this at a museum in Paris at the very first Microchimerism meeting. And I thought, yeah, it's obviously pregnancy evolution in the MHC. I put it on a t-shirt. I wear it to the gym. So in summary, microchimerism is found in peripheral blood in healthy individuals and among diverse hematopoietic cell types, blood cell types. It's also found as differentiated cells in tissues. It's increased in some autoimmune diseases and could be a target of loss of tolerance, but these could also be regenerative in helping repair tissue. May decrease the risk of some malignancies and possibly fuel others. Multiple grafts may be acquired and interactions may have a diversity of consequences. So then there's the good and the bad. And the good and the bad is probably contingent on a number of factors. The source of the microchimerism, the age of the person when they acquired it, the time elapsed since it was acquired, because the microchimeric cell is going to age as well, potential for interaction with other microchimerism sources, and the HLA specific molecules that are carried by it, and also of the person that's harboring the microchimerism and the way they relate to each other. So if that's too many words, I figured that actually popular music has known all of this all along, and so you didn't even really need to come to this talk if you just listen to popular music. And a new meaning to the Beatles song, I am you and you are me. And so we're now acquiring all of the popular music that's actually been telling us this all along, such as there's one in here like Can't Get You Out of My Head. And this one's kind of an inside joke. You heard about that NEMA. There's also an expression called IPA for the inherited paternal antigen. So somebody came up with this, the girl from Ipanema. I thought that was pretty good. And then I think maybe my favorite one is I get by with a little help from my friends. So I really do want to thank all the people that actually made this happen over time, and this is some of the people that worked on it over time, and I mentioned many of their names. Also, just to add that we have a lot of collaborators that really were key in each aspect of the work. And of course, we wouldn't be anywhere without our funding sources, especially the NIH, but also the Scleroderma Foundation, a small grant from the Washington Women's Foundation, and from the Wong Foundation. And so I'd just like to conclude by saying that even if we don't listen to musicians, if we listen to our poets, they've also been telling us this. So thank you very much. Please.
Is there anything known about the variation in the degree of microchimerism across mammals, for instance, with different kinds of placenta, different inbreeding or homozygosity like cheetahs or Tasmanian devils, marsupials versus non?
It's just I love that question, and I would love to know the answer to it. There's not very much known. Kurt Benersky, who was really like kind of the father of the placenta, told me that I should go work on the kangaroo as one of the first of these, and maybe somebody would be inspired to do that. I would love to know the answer to that. There's very little known. There's of course some known in the mouse, but that's not, it's different, but it's in the same categorization. So, yes.
Can you wait?
I find it really interesting that it's class 1 molecules aren't the mediators of the immune response, because I would have predicted that T cells, which are recognized class 1, are the cytotoxic T cells.
Yeah.
But if it's CD4, then is there autoantibody mediated cytotoxicity or CD4 cytotoxicity? I mean,
Yeah.
could you comment on that?
Yeah. This is a good question too. I actually did not expect, I expected to find it across class 1 as well when I proposed this hypothesis and looked at it. And I wasn't unhappy. Like I said, I'm usually wrong. It must be that there are different regulatory mechanisms. And it's interesting because it could have been different. There isn't a lot of evidence that the autoantibodies themselves are pathogenic, other than with a few exceptions in other diseases, like in lupus, the anti-DNA antibody in kidney disease is thought to be pathogenic, and the antibody in autoimmune hemolytic anemia. But most of the rest of our autoantibodies are actually markers of the disease. So I think we actually don't know mechanistically what's going on. We're trying to launch up so we can actually do some functional studies with these cells, and we've managed to achieve at least a log-fold enrichment to be able to do that. But they're challenging. So, thank you.
Thank you. A very provocative series of studies and hypotheses that challenge conventional wisdom, which makes it really great. I may have missed a couple of crucial things here, so I'd like to clarify. One is when these foreign cells first enter the mother's bloodstream, why are they not recognized as foreign and attacked by the immune system? And the second was somewhere in the middle there you said that when the mother has the child cells come into the mother and replace the grandmother's cells or something like that. So what would make them target specifically the grandmother's cells? I mean, it would seem that they'd have to have some sort of recognition function, and that doesn't seem plausible.
Mm-hmm. Well, on the first question, we do know that there is a regulatory T cell response to the fetal cells by the mother, and also in the other direction, that there's a regulatory T cell response of us to our mother's cells. The answer to your first question is a little more complex because it's different, I think, during pregnancy than after pregnancy. There's actually not a whole lot of intact cell transfer, I don't think. I mean, it happens, obviously, but by far and away from the fetus to the mother, the vast majority of what's being found in the mother's blood is actually cell-free. So it's probably packaged in apoptotic, in blebs, and sloughed off the placenta. So there's very, very high amounts of fetal origin cell-free material, like in the 3rd trimester, it's more than 10% of all serum cell-free DNA. And Hilary Gamble tells me it'd actually be quite a bit higher than that. So I think the mechanisms are different during pregnancy than the legacy mechanisms. But clearly, for whatever reason, we have a regulatory response, and so we actually do tolerate these cells to a certain level. Now, why would the fetal, as I said, we often do not get too confused. We try to call the woman in the middle of the proband and talk about the child of the proband and the mother of the proband. So why would the cells from the child of the proband attack the mother of the proband cells and not attack the mismatched mother? I think that's a reasonable question. They're going to be, it gets a little complicated because there's a variety of different HLA relationships. There's the potential for complete mismatch across those generations because it's not necessarily the grand maternal haplotype that got transmitted through to that kid. But I don't know that we know the answer to it.
My question is sort of related to the last one.
Yeah.
It's about what happens, after birth during that sort of immune clearing phase where a lot of fetal cells get removed, and also at the time when involution occurs after lactation and there's an immune process. Is there anything known about what is going on there in terms of both the fetal cells and potentially sort of leftover maternal cells or grandmaternal cells at those 2 time points?
So I think the postpartum window is, to me, one of the biggest black boxes. I really have a hard time believing. I have never seen a grant on that, and I've never seen a paper on it. And yet, that is a period of extreme vulnerability to a whole host of autoimmune diseases. It's known there's an increased incidence of new onset rheumatoid arthritis. It's known that people who have rheumatoid arthritis flare postpartum. It's known that people who have multiple sclerosis flare postpartum. Even lupus patients who don't get better flare postpartum. And nobody's really looking at the immunology of that window. I think it's fascinating. If you look at Diana's original paper in 1996, she described 8 women that had given birth to sons, and then the long-term persistence of male progenitor cells up to 27 years later. The 2 where she didn't see it are the ones where within a year of birth, which I always find very interesting. And I have this question, are they going somewhere? Are they going to the liver? Is the liver a tolerogenic organism in some way? Because the liver is the thing that ignores HLA. Everything else cares. Every other kind of transplant cares about HLA. The liver almost prefers mismatch. So I don't know, but those are some thoughts.
And the involution after breastfeeding is it?
Yeah, there is a little bit of literature around breastfeeding. There's good experimental literature by a guy named Will Burlingham. And I know at least in one of his papers it suggests that in order for there to be continued presence of the maternal microchimerism, the breastfeeding was correlated with that. But I actually don't know that literature very well.
We have time for one more question. Although we've asked quite a lot, so with that, let's thank our speaker.
Thank you. Thank you for your attention.